All About Dicer-Like 3 by Jen Velazquez, Matthew Noak, Kurt Wendt, Patrick Murphy, and Palin Cow. With me, your host, Felix. Woo!
Did you know microRNAs are endogenous small non-coding RNAs of 18 to 25 nucleotides in length?
Not only that, they negatively regulate their complementary mRNAs at the post-transcriptional level in many eukaryotic organisms. No way, guys! And also, a large number of miRNAs have been discovered in both plants and animals.
That's pretty cool, Felix.
Yeah, push, I know. These dicers cleave pre-miRNAs like this. You got a cut here, a cut there, a whoop-diddy-dip-diddy-doo. Bing bong.
However, miRNAs are not single-stranded and do not have exact complementarity to the target mRNA sequence. Therefore, a single miRNA may repress translation of hundreds of similar mRNAs.
The DCL-3 gene encodes for a dicer protein, which is an endoribonuclease that participates in RNA interference activity. RNA interference is an antiviral mechanism used to repress gene expression. Hey, isn't that typically by disrupting specific mRNA molecules to prevent translation into proteins? DCL-3 utilizes TGS.
Generally, DCL-3 dicers cleaves lengthy double-stranded RNAs into short double-stranded small interfering RNAs, about 20 to 30 nucleotides long, that binds to the target mRNA specifically with the perfect base pairing.
What we have here, folks, is a picture of DCL-3 cleaving the double-stranded RNA. Isn't it so magnificent? The siRNAs produced by DCL-3 elicit transcriptional gene silencing by using RNA-directed methylation pathways.
This pathway is first detected in plants infected by RNA pathogens, in which involves siRNA, core RNAi proteins, polymerase 4 and 5.
The mechanism of TGS-RDDM gene silencing by DCL-3.
1. Polymerase 4 mRNA transcripts are copied into dsRNAs by RDR2.
Isn't he from Star Wars?
No, Felix, that's R2D2.
2. These dsRNA are processed by DCL-3 dicers into siRNA.
3. siRNAs are exported onto the cytoplasm and loaded onto AGO4.
AGO4 re-import the siRNA into the nucleus.
5. siRNA coupled with AGO4 targets and base pairs with polymerase 5 nascent mRNA transcripts.
6. This interaction of siRNA and mRNA transcript recruits DRM2 DNA methyltransferase to methylate cytosine nucleotide on the DNA.
7. This results in transcriptional silencing of transposons and other repetitive DNA transcribed by polymerase 5 and their nearby protein coding genes.
Methylation of the transposon leads to silencing of the nearby coding region. That's right. Gene 1 is silenced, meaning no proteins are made from this gene, while gene 2 and gene 3 are being expressed. Cool! Biological function. The main and first biological function of RNA silencing was as a defense mechanism in plants. DCL2 and 3 produce 22 and 24 nucleotide viRNAs respectively when DCL4 is limited in activity.
It appears, though, that only 22 nucleotide viRNAs are sufficient to mediate independent antiviral silencing.
Sorry!
Felix!
Now we're going to look at DCL3 in action with a study called The Identification of RNA Silencing Factors with a Potential Role in Plant Defense Against Pathogens in Arabidopsis thanalia.
The plant's mechanisms for viral pathogen have already been confirmed by research, but its mechanisms for non-viral pathogen defense still remain uncertain. In this research, a root-infecting fungus is used as a pathogen to investigate whether RDDM pathways play a role in the defense of Arabidopsis against non-viral fungal DNA.
The following are their findings.
The end. Say goodbye, Felix.
So long, suckers!
Free created using Powtoon.